Okay. So for those who don't know, clinical trials are experiment studies done in trying to understand the efficacy of some new treatment, like a new vaccine or something like that. And a key part of clinical trials is recruiting patients into that trial. And that's often a major impediment, right? So you need sufficient patients and the right patient population in order to properly run this experiment. The use case, because of that, there's a need to kind of match a cohort of patients with a set of trials. So let's kind of think of this problem purely from an IR perspective. You might have a patient or query. So I'm going to use patient and query interchangeably for the purpose of this talk. And then you want to try and you've got a whole bunch of clinical trials there. And just to give you a bit of an idea, this is an example trial from one of 300,000 at clinicaltrials.gov. It's kind of like a semi-structured document, title, description, etc. And so what you want to do is, given this patient, you want to generate a ranking of trials such that you can recommend the most likely trial that that patient could enroll in, okay? And then, of course, if you've got a bunch of patients, then you have obviously a whole set of queries there. And that's essentially what TREC, Precision Medicine and Clinical Trials track, to which we participated. That's the exact model that they used. But if you sort of stop and think about it a bit more, you realize that this cohort of patients are, not necessarily independent of each other. So let's say they all come from some particular breast cancer clinic. Clearly, they're going to have shared common conditions. So there's some dependency between the patients/queries. And as a result, you're obviously likely to have trials retrieved that are common to the whole set of queries. So how does this play out? Let's look at a little example. You have a bunch of trials and a set of queries or patients, and the X marks whether that trial is relevant to that patient. So now let's say we have a strict cutoff in terms of a number of trials, and that's actually a real constraint. So let's say the same small breast cancer clinic may only be able to be affiliated or run 2 trials, right? So in that case, you'd only be able to cover 2 of your patients. Now, what happens if we were to instead think about choosing our trials such that they maximize the coverage of your patient, right? So if you were to suggest these 2 other trials, then you could enroll or recommend all your patients to at least one of the trials. So where before we had this single ranking system or single ranking for each query, the whole idea here is around cohort retrieval. So it's around generating a ranking of clinical trials for the entire set of patients. And there's actually a number of benefits for this kind of cohort-based approach to the problem. Firstly, to the patient, right? So if you're able to enroll those patients into trials that you couldn't before, they now have access to the newest treatments, and in the cancer space, they can actually be access to life-saving treatments. From a scientific perspective, you're now enrolling more patients into trials, better patients into trials. That means the trials are more robust, more rigorous, experiments being done, and essentially you're advancing medical science and research. And then finally, clinical trials is a major cost of the entire kind of treatment development, drug development pipeline. And so if you can streamline that, there's major advantages. But also patients are compensated for participating in trials, and clinicians are reimbursed for time spent recruiting patients into trials. So there's multiple benefits from a financial perspective. Okay, so what did we do in this ADSCS paper? So as I said, we kind of broke from the idea of treating queries as independent of each other, and we looked at generating a single ranking for an entire set of queries. We then proposed this recruitment coverage evaluation measure to capture the quality of a cohort-based retrieval method in terms of success in recommending patients to trials. Then we looked at a couple of cohort-based retrieval methods, one by adapting rank fusion, and the second by a diversity re-ranking method, and we evaluated those. And then we sort of thought, okay, well, what does an optimal solution look like in this space? And we implemented this greedy set cover measure in order to provide kind of like a computationally feasible upper bound for this problem, and also to kind of tease out some of the underlying characteristics of cohort-based retrieval. So, methods. So first of all, we needed to come up with a measure of success. So that's the evaluation measure. So as I said, really we care about what portion of our query set was there found to be a relevant trial. So 2 out of 5 here. And in this case, you're covering 4 patients. So you only really care about finding one trial that's relevant for the patient. Doesn't really matter if you find more than one. So plugging that into an actual measure, we have this recruitment coverage measure here. It takes a ranking of clinical trials and a set of queries. And essentially we're saying what portion of the entire set of queries are covered by this trial, right? So 4 out of 5 in this case, 0.8. Okay, now we started off looking at, okay, what's the traditional TREC style approach to this? And we used the 2017-18 precision medicine track. And we did a standard BM25F baseline. And then we wanted kind of what's the state of the art here. And we took the 2 best performing systems at TREC 17 and 18. So those are individual rankings for each query. Okay, then what we did is we looked at adapting rank fusion for this problem. So just as a reminder, in rank fusion, you will be taking for a single query, multiple, say, system runs, and fusing those together to form a single final ranking, right? So what we did instead is we took the individual rankings for each of the queries and fused those together to form the final cohort-based ranking. And we use 3 popular fusion methods here with score normalization. Okay, but then, okay, if you think about that, then in this ranking, trials that are very similar to each other are likely to be close to each other in the ranking, right? And that's not necessarily advantageous because 2 trials might just cover the same set of patients. They may be relevant only to the same set of queries. So you're not improving your coverage by having these 2 trials that are really similar. Okay, so that's obviously a diversity problem, right? So what we looked at is re-ranking that initial fuse set with maximal marginal relevance to diversify the ranking and hopefully cover other queries that we hadn't covered before. So let me explain how that works. So you have here a bunch of trials, a ranking of trials. And for each trial, we represent the trial as a vector, which is a distribution over the query set. So essentially saying just that this trial was retrieved by this query. Okay, and then the way the ranking proceeds is, say we're at rank position 2, and we want to then rank the next trial. What we'd like to do is pick the next trial such that it's somewhat diverse from the previous. The way we do that is the previous trials are accumulated into this previous vector together, and then we do a comparison with all the candidate trials to be ranked according to how much they diverge from the previous trials. So we use Jensen-Shannon's divergence to do that. So that's your diversity score. But then you also want to kind of control or contrast that against the actual retrieval score, which came out of the fusion method before. And also, so we want some control about how much diversity we apply. And so we essentially, we have this mixing parameter alpha of the 2 that mixes the 2. And then of course you proceed, you look at all 3 and you choose the next trial that maximizes this function. Okay, so that was fusion plus diversity re-ranking. And next, we sort of thought about it a bit more and said, okay, well, how well can you do in this situation if you were to optimize a ranking for this recruitment coverage? And a true optimal solution is basically the power set of all the trials. So that's NP hard. And we instead looked at this greedy set cover implementation. And this is really done as a comparator. So I'll explain this with an example. It's the easiest. So you have 3 trials here. Now R here represents whether that trial is relevant to that query. So we're using the QRLs here. And in greedy set cover, essentially you proceed by choosing the trial that maximizes recruitment coverage. So we choose the red trial, it's relevant to 4. Then we choose the yellow trial because it covers that blue patient who wasn't seen before. And then, well, this trial isn't actually even needed because we've already reached full coverage with the first 2. That's greedy set cover. Greedy naive is different in that it basically just purely ranks based on the number of queries to which that trial is relevant, right? So same set of 3 documents. You choose this one because it has 4 queries. Then you choose the blue trial, noting that this doesn't improve coverage at all because it's fully overlaps with the red, and then the yellow, right? Okay, so those are just used also to kind of, with the greedy naive gives us a little bit of understanding of, is this overlap situation something that we see a lot of, and does that impact coverage a lot? Okay, let's look at the results. Okay, first of all, the single query ranking results, so the baseline and the best systems that track. So really the thing to note here is the big difference between the 2. So someone did a very good job at track in this task, and you can see there's a large difference in effectiveness. Now, when we saw this, we thought, oh, okay, well, I would assume that then directly when you're fusing those into a single ranking, you would again see a big difference in the next task. But that wasn't actually the case, which was somewhat surprising to me at least. So these are, on the y-axis is our recruitment coverage, right? And on the x-axis is that rank cutoff, so the number of trials that you could recommend. And what we see is for the score-based fusion methods, no real difference between these 2 initial rankings. So even though here, big difference in the individual query results, but when the fusion is done, you're not really seeing much difference. Okay, reciprocal rank, you do see a difference between the 2. But it's important to note too that if you consider just a small number of trials to recommend, which is actually a sort of a real use case. Remember before I was talking about 2, 3 trials being recommended to someone. There, the actual far less effective BM25 initial rankings actually performed better. Okay, so remember then we take these fused results and we apply diversity re-ranking, okay? So this is the same plot. I've just chucked on the diversity re-ranking as the red lines. So here we do see a difference between the solid line using BM25 initial rankings and the best Trek teams. So the diversity re-ranking is actually there, in this case is exploiting the higher quality Trek best initial rankings, okay? Except for our troublesome 3rd child reciprocal rank, where we're not seeing much difference there. Okay, so now let's look at the greedy methods. So once again, same plot, I've just added on the 2 greedy methods here as well. And there's a few things to note. I guess, first of all, the greedy naive approach was pretty poor, right? It's well below even these fusion methods. And what that is telling us is that there's high overlap in terms of the trial being relevant to the same set of patients, of the same queries. And that is, well, I suppose two things. One, it's motivation for the diversity re-ranking approach, right? And the second one is that there's obviously dependency or similarity between the queries, and that kind of motivates this cohort-based approach where you can optimize a final ranking for the whole set rather than for individual queries. Okay, the next is clearly there's obviously work to be done. I mean, we're not reaching full coverage even when we're right down here at 100 trials. And certainly if you're only able to recommend a few trials, then you're pretty poor. So there's plenty of fun work to be done ahead. Then if you look at greedy set cover, right? So in this case, and I think there were 80 or so queries in total. So you're able to reach full coverage for 80 queries with just 30 documents, with 30 trials, right? So if you were to, and 30 trials is really easily within the realms of, say, a busy clinician being able to review trials. So if you were to get these methods closer to this, you actually got a really effective search or recommender system in terms of taking a cohort of patients and getting a small manageable subset of trials that you could refer them to. Okay, next is we had a look at the impact of this diversity mix, which is, if you recall, when we do the diversity re-ranking, we trade off diversity and the retrieval score. And probably just thing to note here is that the diversity was helpful, right? In all cases, applying some amount of diversity led to some improvements. But also, again, you see the situation whereby with a diversity re-ranking, you see a much bigger difference with the high quality initial rankings from the best TREC systems. Now, in the paper itself, we actually go into, so here we've only got 2 initial rankings to fuse. In the paper itself, we looked at actually generating a whole bunch of synthetic rankings of set precision levels, and to try and understand, well, what is the impact of the quality or the effectiveness of the initial rankings in terms of the downstream recruitment coverage. So if you're interested in that, it's all covered in the paper. Okay, so what did we learn? I thought we learned quite a bit. The first thing which I thought was a bit surprising was that the initial rankings didn't always translate into really good recruitment coverage. But when we had that approach of fusion plus diversity re-ranking, then we were actually able to see some results that I think would make this approach usable or valuable as an actual trial recommendation system. And then I suppose as we got further in, we started thinking about, okay, well, what happens if we had specific cohort-aware ranking methods? So we'd always approached it as, okay, you take these initial runs, you fuse them together, you come up with a final run. But we saw there that there's, that doesn't take into account, well, the diversity takes into account whether trials are similar to each other, but we don't really care whether a trial is similar to each other. We care whether it captured a different portion of the query cohort. So you can imagine a kind of cohort retrieval method that would capture things like patient similarity, which is query similarity, similarity between trials, predicted coverage, basically thinking about how can we develop a method that is tailored specifically to ranking those trials for a cohort of patients. And I think that would be fun from a computational computer science perspective. I said profitable here, but really I mean, I think it definitely has some real world applications. There's certainly a major issue in matching patients to trials. And if we were to get this right, I think you could definitely improve that process. And if you were to really develop a system that was quite accurate at recommending trials on a large scale, you are really allowing patients access to potentially life-saving treatments, particularly in the cancer space. Thanks very much.